is that he's just so humble. When he comes up here, you'll be amazed because he's just got a pile of accolades. And I'll read some notes here that we have, but he's an amazing person. Professor Philip Marsh is a microbiologist based in the UK who's been involved with oral microbiology for most of his career. And one of the things he said to me is, at times he was wondering, what's it all for? Until he came here and he started realizing that we're looking for practical applications for everything he's spent his entire life doing. He has developed novel ideas on how dental plaque exists with the host in health and disease. This research has resulted in numerous awards. In 1991, Professor Marsh received the Orca Research in Dental Caries. In 1998, the IADR, the International Association for Dental Research, Distinguished Scientist Award. And recently, in 2001, the Society for General Microbiology was based in the UK for applied research. So without keeping you any further, I present to you Professor Philip Marsh.
Thank you very much for all these warm words. In fact, I've never been so nervous in my life before I talk because I've had so many complimentary comments. And it's absolutely true in that I'm a microbiologist, so the one thing that frightens me is no mention of patients in this. This is looking at dental plaque and trying to understand how it has this complex relationship with each one of us in health and disease. Because plaque, as you will see in my slide, is natural. We all have it. You'll never get rid of it. And in fact, it does us good. You can't live without organisms in your body. At the same time, it gives you all a job, because it can do mischief, it can do disease. And it's understanding that relationship, because dental diseases are different to many of the medical diseases that you'll hear colleagues talk about. So I spend some of my time now working on tuberculosis. In general, you have to catch the organism from someone else. It gets in the lung, it shouldn't be there, and it causes disease, and you treat it with a vaccine or antibiotics. It's a very difficult disease to treat, but at one level, it's very simple to understand. It's an overt pathogen. What we're talking about with dental diseases is something that's much more complex and much more complicated to understand. And in my lecture, I'm going to try and explain how I feel I came up with a much simpler way of understanding the relationship between these bacteria that we all have and we should cherish, and why we get disease in some people and other people stay healthy, and how you can exploit that knowledge to try and keep people healthy while maintaining the benefits of these organisms. So first of all, just to let you know where I come from, the small island off mainland Europe, the United Kingdom, and I have a sort of more complex lifestyle in that I work in two places. So most of my time is spent at something that's newly set up in the UK called the Health Protection Agency, which is supposed to look at microbiological, radiological and chemical, natural or deliberate threats. So we have laboratories all over the country that can look after the most nasty organisms that exist. And I've done oral microbiology there, and I'm now doing tuberculosis research there. Then I travel once a month via London up on the train to Leeds, which is in the north of England, where I have my professorship and I teach and do research in oral microbiology. So this is Salisbury, where the nearest city to where I'm based, and in the south of England. It's a medieval city. It's a little bit older than Chicago, and it's famous for the cathedral, which has the tallest spire in England at 404 feet high. And it was built in 1230, in about 30 years, which is quicker than some of the builders I have for my house. And nearby is an even older structure, about 10 miles away we have Stonehenge, which is where we learn to play soccer with these ancient goalposts. When I get to Leeds, Leeds is an northern industrial city, but it's been fortunate in retaining a lot of its Victorian architecture. So these are some covered markets, and this is the university, or part of the university, that's up top of the hill there. And I should acknowledge at this point that the work I'm going to describe that led me to come to some of these ideas and hypotheses is really due to the brilliant work of some of my colleagues. So I need to pose you some questions. I need to engage. And I would say that so far I've paid all my own travel, hoping to get it back. But I noticed there are score sheets in the pack. So unless you give me a decent score, I may not get all of this back. So one thing was engaging the audience. So this, you can tick this box now. So does dental plaque play an important role in your professional life? No? Don't hear anything? Yes? We can help. Microbiologists know a lot about dental plaque. There are lots of bacterial types. We found more. We've given complicated names for you. You can impress your patients. We like to change these names fairly often so you don't understand it. And we'll even offer you Bug of the Month to help. Is this interesting to you or helpful to your patients? No. what I'm proposing and trying to convince you of in this lecture is that understanding some ecological principles may help. It may simplify all of this complexity into ways that you can meaningfully translate to helping to improve the dental health of your patients. So the structure of the talk is I'm going to briefly just review the relationship between all of us and our microorganisms, give you a contemporary view of dental plaque in health and disease and use the biofilm word, describe how there is a dynamic relationship between the environment in the mouth and the body in general, and the composition of dental plaque. And that's just really understanding the basic principle. I think that changes in the mouth will be reflected in changes in the oral flora. Explain how ecology can be applied to this, cover some implications for disease control, and then summarize. But first of all, again, I've got to pose some questions to you. We have a television program, or we used to have a television program in the UK where you have to spot the odd one out of 4 similar-looking events. So first of all, you can spot which is the odd one out. Is it the devastation of forests due to acid rain? This is Scandinavia, where all the outputs from the UK power stations gets deposited there and ruin their vegetation. The overgrowth by algae on lakes and streams and rivers due to inorganic fertilizers, nitrogen phosphate flowing off the fields, goes into the water. The algae use those fertilizers, they grow up, they consume all the oxygen in the lake and kill the aquatic life there. The loss of dinosaurs when a meteorite impacted many hundreds of millions of years ago. In fact, I was very lucky to get an early photograph here just before the meteorite impacted, and Diplodocus is going to be in trouble, or dental disease. And my view is that they're all due to similar principles. They are all due to ecological changes that have a cause and an effect, and therefore by understanding it in ecological terms, the whole problem becomes much simpler. So starting off with the relationship between man and microbes. It's been estimated that a human being is made up of about 10 to the 14 cells, of which only 10% are mammalian. So the rest of those cells, the majority, the 90%, are actually microbes that inhabit all of the environmentally available surfaces of the body. We all have this microflora on our skin, mouth, gut, and so on, and they are of benefit to the host. It's been shown by studying germ-free animals. These are animals that are reared under abnormal conditions where they're never exposed to microorganisms, that in the absence of a normal flora, you don't get the natural development of the physiology and host defenses in those animals. So there is a direct relationship between the organisms that naturally inhabit us and our natural physiological development. It's also been shown that this normal flora on any surface in the body is important to excluding microorganisms we come in contact with daily. We don't inhabit a sterile environment. We come into contact with organisms in the air, in our food, water, and just meeting people. And in general, we don't get colonized by these organisms because of our natural flora. And this applies to our resident oral flora as well. And the sort of principles that apply is that the organisms that we naturally have that have evolved with us over millions of years is that they are far more competitive sites at which they can attach. They can be much better at obtaining essential nutrients. These are usually derived from the host. So the organisms that the normal flora live off are those usually provided by the host. So for example, in saliva, there are proteins and glycoproteins that normal flora live from. They create microenvironments that are disadvantageous to exogenous species and can also produce inhibitory substances that will again discourage these transient organisms from establishing. And something that's happening currently is trying to understand how the body can recognize and tolerate these massive numbers of commensal organisms. As I say, 9 out of 10 cells, 9 are microbial, 1 is mammalian. How they can recognize these organisms and not react negatively against them, and yet still respond to pathogens when we need them. So people have looked, for example, at the gut, where there are huge numbers of microorganisms lining the gut, have shown that when organisms attach to the surfaces of the epithelial cell lining the gut, you get transcription of pro-inflammatory genes, so that you will get an inflammatory response, very undesirable in the gut. What's come through now in a series of studies is that there is possibly crosstalk between our commensal organisms and the host. We say, I'm really good for you, don't respond to me. And this process downregulates this pro-inflammatory response while retaining the ability to recognize a true pathogen and then be able to give an appropriate immune response. In a paper that's in press, in Infection and Immunity, available electronically from the site, but isn't in hard copy yet, has been done by a colleague of mine at Leeds, Deirdre Devine, and a colleague in Canada, that has shown that Streptococcus salivarius, which is a very common oral mucosal organism, can downregulate innate immune responses in human epithelial cells. So we're now beginning to realize that it's not just we have a barrier of organisms, say, in our mouth or in our gut that wards off pathogenic organism, but it's actually a much more active and dynamic process. By having these natural organisms that evolve with us, they're actually signaling to the host, and there's a dynamic relationship between the host and these organisms. It leads to the uncomfortable conclusion that we made more microbial than mammalian. So if you look at the distribution of these organisms, as I say, every site in the body has microorganisms associated with them. And if you look at the types of microorganisms, which microbiologists like myself will sometimes do, there are differences in the organisms at each site, which means that the habitat can select. We swallow about a litre and a half of saliva a day, and yet, so saliva contains about 100 million bacteria per mil, and yet very few of those microorganisms, they pass through the digestive tract, ever get established in the gut, yet they have every chance to do so. And that you can only conclude is because the environment in the gut is distinct, that it selects for a subpopulation of organisms. If we look in the mouth itself, there are distinct surfaces. There are obviously hard surfaces associated with teeth, mucosal surfaces. And again, if you look carefully enough and identify the bacteria to a sufficient level of discrimination, there are differences in predominant types of organism. So we find organisms like the mutans streptococci more commonly on teeth than on mucosal surfaces. Again, even though they get present in saliva and theoretically colonize all the surfaces. So again, we see that properties of the habitat, even within the mouth, can select for certain types of organisms. And if we look further in more detail on the tooth surface itself, and here I've just distinguished Fisher plaque from gingival crevice plaque, you find substantial differences in the microorganisms. So, for example, in Fisher plaque, it's mainly gram-positive bacteria, such as streptococci. You find very few gram-negative organisms, whereas if we look in the gingival crevice, we find it's the predominant site for the obligatory anaerobic bacteria that use proteins for their nutrition, whereas these tend to use sugars for their nutritional sources. And this, again, reflects differences in the environment. The only conclusion you can come to, the change in the environment here means that some organisms will do better and others will do worse. And if we look at the site, we can see there are differences in pH. This is much more saliva influenced, whereas here in the gingival crevice, I apologise for the people on this side of the room, we have gingival cricula fluid that bathes this area. And apart from bringing in components of the host defences, based on more serum-like exudate, so IgG and neutrophils, it also brings in a range of different proteins and glycoproteins that happen to be wonderful substrates for some of the organisms we associate with periodontal disease. So the anaerobes love this environment. We provide just the sort of food that they love to grow. So we conclude that, again, the microhabitat selects. Because these organisms have every chance to colonize every site, but they're localized. So we can conclude this point, that the habitat selects which organisms are best able to grow there. Habitat is also hostile. Not every organism can grow in every site. So there is a direct relationship between the environment and the organisms that grow. And this is true of the mouth, the gut, and rivers, streams outside. Now we're going to get a bit heavy for a while because we're going to talk about dental plaque. And this is sort of some sign that reflects our current understanding of complexity of it. The definition of dental plaque that I use is that it's a community of microorganisms found on the tooth surface. An organism on any surface, and that's where they're normally found in nature, are now referred to as a biofilm. It doesn't mean that plaque suddenly changed in any way when we use the term biofilm, and see it in all sorts of contexts. It means we're more likely to get funding money if you use the word biofilm. And it also means we can extrapolate about things that are happening in the mouth, because principles that apply to a biofilm in a cooling tower, in a catheter-associated infection, or on teeth could be broadly similar. So it does have advantages considering it as a biofilm because of the ability to utilize information from other sources. So it's a community of organisms embedded in a matrix of polymers as a biofilm. And these polymers come from saliva, they come from the host, and they're also from the bacteria themselves. The significance of this word community, which is often not used about dental plaque, it wouldn't matter if the properties of the organism in dental plaque were merely the sum of all the individual cells. And that being a biofilm, the properties were the same when they're on surface as when they're free living in solution. Important of these two words is that when these organisms are together as a community, you get more than the sum of the properties of the individual organism. They can do things that they can't do individually. It's a bit like this group of people here. You can achieve things that perhaps would be more difficult for you to do as an individual person, but collectively you can influence and achieve additional benefit. Likewise, it's now known that organisms, when they attach to the surface, change their properties. So they downregulate certain genes and upregulate others to allow them to be successful in their new environment. So these are two important words. I also wish to stress, as I said in my introduction, the black is natural, normal, and has benefits for the host. You will never eliminate it, you should never even try. What you want to do is to control it and keep it at levels that are compatible with health. So I'm just going to talk a little bit about biofilms. They're ubiquitous. It's the natural place where microorganisms live. They generally rarely live in liquid suspension. Although you can find them there, it's usually because they've been washed off a surface. They can act as haven for pathogens. So many overt pathogens can survive within the complexity of a biofilm because they hide from the host defenses. And as it says here, they have altered microbial properties. This is biofilm in a water distribution pipe. These are biofilms that have been lifted from machinery. So the problem with a lot of machinery is they eventually slow down, grind to a halt because microorganisms get in the oil, the cooling systems, and they just love the surfaces that grow on there, and eventually the thing fails. Any people wearing contact lenses today? Microorganisms like contact lenses. These are hip replacement joints, and again, a fear with putting any implants into a person is that microorganisms will attach those, grow, and set up a chronic infection. Here's the contact lenses. Biofilms can establish on contact lenses. Honestly, that's less desirable to put in contact with your eye than a nice clean lens. Legionella hides in the biofilms in hot water systems, and we have plaque itself. And again, I stress that just because plaque is now called a biofilm doesn't change what plaque is, but it does mean we can understand from events that occur across the whole of medicine and microbiology about what we may know about dental plaque itself. We can apply that knowledge. So we begin to learn over the last 5 years or so a lot more about dental plaque biofilms through the application of new techniques, new imaging techniques, and so on. So imaging, big step forward for studying biofilms, has been the application of novel microscopy techniques such as confocal microscopy. In the past, we've used electron microscopy that shows biofilms to be very compacted, dense film. What I'll show on some slides is that when you have confocal microscopy, you can view biofilm in their natural liquid state, so they're still covered in liquid. The process for electron microscopy means they have to be dried down and the biofilm's compressed. And there's a lot more space and movement for molecules within biofilms when you view them in a natural state. It's now possible to use molecular techniques to identify and recognize, detect even the most fastidious microorganisms. The organisms that we can't grow, we can now recognize they're there and they're probably doing something. Just because we can't grow them doesn't mean they're not important. People now begin to recognize that there may be virulent clones of certain periodontal pathogens. So some in what used to be called juvenile periodontitis, there does appear to be a very pathogenic clone of an organism that has the nickname AA that comes from northwest Africa and has spread into other countries and seems to increase the risk of having very aggressive periodontal disease in adolescents by sort of 15, 18 times. And again, now that genomes are being sequenced for some of the oral organisms, we can begin to understand more about what they're doing when they're living on teeth, when they're causing disease, or being treated by antimicrobial agents. I'm going to go through some of the stages in how these dental plaque biofilms form. These stages I'll go through 1 to 7 fairly quickly, but I would just say that they're more for convenience. In reality, these phases are constantly happening. You don't have to get finished stage 3 before stage 4 happens. It just makes it easier for me to subdivide and explain. So if you clean a tooth surface, within seconds you get components from saliva being absorbed onto the surface called the acquired pellicle. Microorganisms are transported passively by the flow of saliva towards the surface, where they can be held reversibly by weak long-range van der Waals forces. This can become more strong and irreversible by specific short-range forces between molecules on the cell surface and complementary receptors in the pellicle. And then as these organisms begin to grow, we call them primary colonizers. Secondary colonizers that are unable to attach to the pellicle can sometimes attach, again through specific receptors on primary organisms and adhesives on the secondary colonizers that can attach in specific ways, often through lectin-like interactions, in other words, carbohydrate-binding proteins, those primary colonizers. People are beginning to dissect out these adhesin-receptor interactions. And the importance of this is that it could be helpful to develop analogues that block these very specific interactions and stop certain organisms attaching in such high numbers. So here we have some of the organisms, many streptococci at the start, and they attach to specific molecules in the pellicle. As plaque develops and matures, a great big pointy thing comes down, fused bacterium nucleatum. It has the advantage of binding to these primary colonizers, but acting as a source of attachment to some of the more anaerobic organisms that you associate with periodontal disease. So this is considered to be a significant organism in the development and maturation of dental plaque. So it builds up into three-dimensional film. The organisms are in close contact with one another, so they can interact metabolically. They start to modify the environment. So you can pick out of dental plaque organisms that are so fastidious in either their growth requirements or their sensitivity to oxygen that even in the best lab in the world, we can't grow them. We can detect them through the molecular techniques. They're modifying the environment so it suits them. And within it, you get gradients, and they synthesize polymers to make this a very sticky, difficult to remove biofilm, and also to help wall out molecules that we might try and apply to influence them. We now know as well over the last few years that these organisms don't just interact metabolically through chemicals, they can signal to one another and say, hey, I'm here, I'm next to you, we should start to do some trouble. So you get gangs of these organisms that start to signal to one another, say there's enough of us now to cause mischief. So again, people are trying to understand these signaling mechanisms. It isn't just unique to dental plaque, it applies to most biofilm infections, because if you understand the language the organisms are using, you can perhaps tell them a lie and tell them to attach because conditions aren't good. So in cystic fibrosis, the organisms in lung form biofilms, they make different signaling molecules, but as a therapeutic, people looking to see if they can give them a signal that says, this is a very nice place, but you want to move on somewhere else, and stop them forming biofilms. So again, this is the way you understand how things happen when you study biofilms in different circumstances. The sort of standard biochemical interactions in biofilms can include the development of food chains and food webs. Bacteria share enzymes, so if a substrate comes along that they want to use and they can't do it, they'll combine with a friend and say, you've got this enzyme, let's work together and make this happen. This is what I'm saying about community effect. This is a community process. They signal, they can combine to inhibit inhibitors, including antibiotics, and they can transfer genes. So if you look at this food chain, food web concept, many organisms will convert dietary carbohydrates to lactate and to other fermentation products. But these can also be consumed by other organisms. So for example, the classic one is Villanella, gram-negative coccus. It can't utilize carbohydrates directly, it doesn't have the right enzymes. It lives on lactate. So you get these food chains where lactate actually doesn't exist for very long if you have lots of Villanella nearby. It gets converted to acetate. And other bacteria in these complex units can in fact take these molecules down and convert them even further. So you get these mutual interdependencies, very complex, very highly developed and evolved. So here we have a biofilm again. Here we have a cell that produces one of these signaling molecules. It's picked up by a neighboring cell that simulates it. Hello, there's a friend up there. So we have drug taking going on right under our noses, literally. In streptococci, these are through peptides, for example, competent stimulating peptide. The recipient cell, as a byproduct of this process, is more able to take up DNA. So if the plaque is under attack, cells lysing, when these signaling molecules go out, they're also able to take up DNA much more effectively. And in gram-negative anaerobes, they use a different set of molecules. As I said, they can take up DNA, and this microorganisms, the closest they get to sex. So apart from drug taking, we've also got sex. All I know rock and roll is going on there as well. But these do offer new opportunities for control. And as time goes by, these will be explored, people will be looking to see if they can interfere with these processes. At the same time, there are bullies in dental plaque as well, and these will try and force their way against competitors. So they produce a small molecule called bacteriocins, hydrogen peroxide, acids, and compete for food. So there is warfare going on under there all the time, and violence. It's a nasty place to be. Organisms can sense their environment. They're very sophisticated, and if conditions become extreme in any way, they can sense this and attack. And they do this. Some can produce proteases, enzymes that degrade their adhesion molecule, and so they attach and disappear. So in dental plaque, as I say, we normally look at it through the electron microscope, and it's a very dense and compacted structure. And we interpreted this dense packing to explain some of the properties of how molecules penetrate or don't penetrate into the biofilm. We've assumed that there are gradients, fairly linear gradients within the biofilm. So for example, with food, there'll be more on the outside and getting less as you go down to the depths of the biofilm. And likewise, oxygen, or this is a measure of how anaerobic a site is, the redox potential, that there's more oxygen on the outside and it's more anaerobic, more suitable for anaerobic organisms down in depth. And correspondingly, metabolic products tend to accumulate in the depths of the biofilm and get less near the surface. If we look at pH dental plaque biofilms, the normal source of food for oral microorganisms is the host-derived compounds. For example, in the mucins, they're proteins and glycoproteins. They can use sugars off the mucin backbone. And what we get is a very slow rate of acid production. It's not so easy to get these things off. It's not so easy to metabolize them. And we only get a small fall in pH. And isn't that just how we like it? We keep the organisms there. We don't make much acid. We're all very happy. But as we evolved as humans, we developed sweet taste, like to have lots of sucrose. When we lump that on top of the endogenous substrates, we get very rapid acid reduction, and we can get terminal pHs within a couple of minutes that are capable of demineralizing the enamel. If we look below the gum line, the organisms there can metabolize the gingival cricula fluid and tends to go in the opposite direction. So the organisms that you find in the subgingival environment are proteolytic. They don't tend to handle sugars like the proteins, and they tend to, when they metabolize them, they produce ammonia and other compounds. The pH tends to rise. However, as we're applying more and more sophisticated techniques, this is a very complex microscopy technique that I was involved with. I'm not even sure I can say it. It could be embarrassing if I don't know what it is. It's two-photon excitation microscopy combined with fluorescent lifetime imaging microscopy. And in simple terms, people point a laser at a biofilm, and they flood it with a dye that is sensitive to pH. Its fluorescence is proportional to the pH of the site. You can see here there's a grayscale, and the lighter colors are low pH, and the darker colors are a higher pH. And this was a laboratory-generated biofilm, cultured 910 organisms. And what we found was, instead of having this linear gradient, the organisms create little microzones of pH. They'd be very close to one another, just a few cell distances apart. So they create their own habitat. Which probably explains why we get a small sample of dental organisms on paper. If they read the papers and looked at textbooks, they shouldn't be existing together. They should be incompatible. We have anaerobes in a clearly aerated environment. And that's because within the biofilm, the properties of the biofilm, organisms consume oxygen near the surface. Very rapidly, you get anaerobic conditions. They alter the pH, either making acid, producing alkali, which creates their own little niche. I guess it means when you go poking around those little sharp sticks of yours, you're doing quite a bit of damage to that exquisite architecture. Again, applying this confocal microscopy techniques is to a sample of plaque that was generated on a removable enamel device in human volunteers that we begin to see, although it's probably not as convincing as some of the cartoons you see in papers, but there are channels. Instead of it being a very dense compacted structure, there are channels and spaces that generate within these biofilms. The area that is quite frightening, if anyone of you of a nervous disposition or needs to take medication for heart problem, this is the time, because in terms of knowing what's in dental plaque, I don't know when any of you are taught and may have learned what organisms are there. It's changing very rapidly and very quickly. In the olden days, when I started, we used to take some petri dishes with some food in them that all bugs would grow. We'd have some that would grow, we thought, everything, and some that were selective for only certain subtypes. We put them in a couple of atmospheric dishes and incubate them for a while, and we get the colonies coming on plate, and then we'd pick them off, ramskin them, do a few simple tests. And a couple of weeks later, we'd know what we got. And even when we got quite sophisticated, we could come up with a fairly formidable list. These would be in every sample of plaque. But if you looked at enough samples of plaque from different people, different sites, this is what we'd say you could find in dental plaque at any time. These are what's called bacterial genera, the genus, and that genus is like a family name. So one of those could be the equivalent of Marsh. If you look more closely, you can find within them, most of them have many species. So there could be a Philip Marsh, a Jonathan Marsh, the other children I've got, Catherine Marsh, Thomas Marsh. Same with some of these, there are lots of different streptococci buried below them. Now we're using, it's possible, and again this applies to all biofilms, not just dental biofilms, molecular techniques where you can extract the DNA, you can amplify certain genes with universal primers, you can sequence, look for homology of these sequences in international databases, and suddenly you're realizing there's a whole different, it's like the Amazon in there in terms of the numbers of microorganisms. The benefit is you can generate probes from what you learn and apply those in fairly simple techniques. And I know a lot of you are sorts people, so you go fishing in dental plaque for certain organisms. That's fluorescent in situ hybridization, and flies needed or anything. And you can look for certain organisms. And this is really becoming very challenging. It's recognized that at least half the organisms in plaque can't be cultured, even in the best laboratories in the world. If you just take one group, spirochetes, we're always talking about spirochetes and periodontal disease. We can grow some of them now, I think are very clever. But when these techniques were applied, in one study alone, we found 47 species using molecular techniques that they can't grow. Are they important? Are they critical ones? Or are they just bystanders? We don't know. And there's another group, the TF7, again found in subgingival plaque. It doesn't even have a name. It's supposed to go R at this point. So we've got a group of organisms, which is called the TF7 clade. And again, it's found more commonly in sites with inflammation. Are they causing it? Are they responding to inflammation and growing up because inflammation provides them with nutritional sources? We don't know yet. This is still a very dynamic, changing field. So perhaps if you're on the West Coast in a few years, there might be a chance just to update. So this is the type of list we have now. This is horrific, isn't it? Because again, these are the genus names, and behind a lot of these organisms, there are many species. What do you do with it? Is this helpful? So I said, beginning with microbiologists, look, what's wrong with you? We've given you all the information. Why don't you go and prevent disease? These are the organisms that somehow must be causing disease. Why are you unable to apply this? It's very complicated, but we can simplify it. And I want to just show these couple of pictures because I thought they were quite instructive. This is looking at sample plaque where you use a live dead stain, cow or certain organisms. This is live dead stain. This is showing that the uncultured spices using one of those probes are red. And it's like some of these organisms are just the occasional rare chance occurrence. These can be quite numerous in these samples, and it could be that we're just not detecting critical organisms. This is that TM7 organism shown in blue, and it's exquisite looking organism. You can't help but love the things and carry on to study them. But the big thing that I would say is it's absolutely of no help to people to have all these names. I mean, what does it matter? Or does it matter if someone got centipede something or other? We don't know yet. What we need to know is what these organisms are doing, what their function is, what do they indicate. If you detect this organism, does that mean there is a certain set of events happening? So we're going to have to understand a lot more about what the organisms do and not worry too much about lists of organisms. The other important thing about biofilms that you need to be aware of, again, learning from the general biofilm environment, is that biofilms are much more tolerant of antimicrobial agents than the same organisms growing in conventional liquid or planktonic culture. So if you apply an antimicrobial agent, it often won't penetrate all the way within the biofilm. The organisms produce these slimes, these matrix, and some antibiotics will bind to slime and won't reach, they'll have effect on the surface, but won't reach the organisms below. The organisms, when they start to detect the presence of an antimicrobial agent, often induce a stress response. And so they'll upregulate genes that might help to pump out the antimicrobial agent. Down the depth of the biofilms, the environment is different, and that might be less conducive to the action of antimicrobial agent. And there's also possibly a phenomenon of persistent cells that have been genetically predetermined to be a subpopulation of the group of organisms that are designed to be there when nuclear fallout has happened. They will survive and recolonize and take over the world. The bacteria also grow slowly in biofilms. The slow-growing organisms are more tolerant of antimicrobial agents and can also swap genes. So if we just look at some examples, here are in laboratory models. Here is an organism associated with health, one periodontal disease. If you look at when they're growing as a biofilm, it can be many-fold more levels of the drug and need to kill the organisms when growing as a biofilm than when they're growing simply in a liquid medium. The IC is the minimum inhibitory concentration that stops growth. FBC is minimal bactericidal concentration that actually kills the organism. So data that you get from studies on liquid culture do not translate to when you're trying to treat biofilms. This has led people to suggest we should be determining either the biofilm inhibitory concentration or the biofilm killing concentration or even biofilm eradicating concentration. And if you look at data that's been done for Strep sabrinus, it's implicated in caries, for two dental relevant products, you can see that the biofilm killing concentration was many-fold more than what had been calculated on free-living bacteria that how these tests are normally done. It doesn't mean that these antimicrobial agents have no effect in the mouth, but if you're predicting that they're going to be killing, eliminating an organism, they won't. They'll knock them back, but they won't remove all the organisms. And if we just look at a study where people grew some plaque in the mouth, so this is the surface on which they've grown and had been inserted into the mouth, we get this nice big biofilm. Here we have a green stain that shows live bacteria and a red stain that shows there are some red ones in there. We apply chlorhexidine. You can see the main effect of chlorhexidine is on the surface of the biofilm. So it's not going to eliminate the biofilm, and that's probably not necessarily a bad thing, but it will have some effect on it. Also, beware, particularly in relation to subgingival plaque, that there are many sites where some of the organisms in the plaque produce beta-lactamase, and they can be at concentrations that are sufficient to inactivate the penicillins in gingival crevicular fluid. So they may not be the organisms you think you're targeting. They may be sensitive, but maybe another organism nearby that's of no relevance to any periodontal disease, but they're making an enzyme and they will neutralize and inactivate the antibiotic. This is a community, yeah?
Is there any indication that by doing what we've been doing with antimicrobials over X number of years that we're creating a resistant biofilm to some degree?
It's a risk, but I'm not sure I've seen much evidence for that. I think one always has to be very careful with antimicrobial agents, and often in dentistry we're using molecules that are more of a disinfectant nature, and they will tend to be damaging bacteria in a fairly crude way, and the organism is less able to become resistant to it. But certainly if anyone is using antibiotics, one has to be very cautious, because what the next few slides show is that you can get transfer of antibiotic resistance genes, in this case penicillin binding protein, from an oral commensal to a pathogen. Strep pneumoniae can exist in the throat and the pharynx and the mouth. And genes have been shown to be passed from the organism to that, in fact from pathogen back to commensal. So potential is there for reservoirs of resistance genes to accumulate. And if it doesn't affect dental treatment, it could be passed on to other organisms that could be more significant. And just to let you know, this is more sect happening. So again, just to finish off this section, I've emphasized that plaque is only a biofilm microbial community. It means they can live in a broad range of habitats. So you consistently find obligate anaerobic organisms in a hugely aerobic habitat. They can use food that individually they can't metabolize. They work together. Increased tolerance to inhibitors, as I've just shown by beta-lactamases, and indeed some organisms that are very weakly pathogenic, if they combine forces in a gang, they can cause disease. So abscesses are typically polymicrobial because each individual organism is not very pathogenic, but together they can form quite a violent group of individuals. So here we have our plaque. It's a biofilm, so it alters gene expression. It's specially organized. It's embedded in slime, and then they can talk to one another. As a community, there's lots of different organisms. They can grow in places we don't predict to grow, and they have a more efficient metabolism, and they can be more virulent. So the consequences of all of this is that the plaque has a different phenotype than what we might predict. They coordinate their activities, they're functionally organized, and they're more resistant to the treatment we might think they ought to be susceptible to. However, over time, what we get is organisms growing on our teeth, and we get balance. They reach a level, they've gone through coagulation and so on, and we get a reasonable level of balance. And this is despite some modest stresses. So despite the presence of host defenses, as I said, there seems to be some sort of recognition of one another, with a reasonable sense of diet, although I could define what that is. By age, I can't remember why I've got that on there, probably I was having an age moment. And despite the presence of exogenous species, in general, over time, a reasonable level of measuring what the organs are, they remain pretty stable, pretty constant over time. And that's the important thing I want to stress here. That's not due to any indifference by the organisms. They've just not got in there. It's like a retirement home that sat there. Well, this is really nice. You know, I get a bit of saliva, a bit of food. This is really comfortable. These organisms will respond to any change. If you're immune suppressed, they will detect that and respond. If you alter your diet, they will respond. So this ability is due to the fact there are a lot of interactions between these organisms. They're dependent on one another for breaking down the nutrition, for coping with host defenses. So generally, one organism finds it very hard to break away and dominate under normal situations. So what that means is we get the balance, de- and remineralization and equilibrium, then a bit of acid they make is compensated for by remineralization. There's very little inflammation, very little reduction of gingival crevicular fluid. If you have a reasonable level of oral hygiene and a sensible diet, whatever that is. However, in people like me that don't have a sensible diet, useless to clean my teeth. I've kept dentists in yachts for a long time, and better holidays than I have. As you get carers, you get demineralization, exceed remineralization, and you get inflammation and periodontal disease. So what we're having here is something has to have happened to alter this balance. So you get an overload by some environmental factor that suddenly disrupts this, and we get ecological catastrophe. And rearrangement of the structure of that microbe community, which means you can get outgrowth of previously minor components of what's there. This is what we see in rivers and lakes when we chuck in nitrogen or phosphate fertilizers. This is what we perhaps see in acne when people have hormones that feed the bacteria on the skin, or in colitis. It does happen in dental diseases. If we look at dental plaque health, and it's there, you can find a certain nice consortium of microorganisms that we all have. They be found in all of us. However, when we get caries, we get a shift. That's not the picture we see. We get much higher level of acid-producing, acid-tolerating organisms like Muta, Streptococci, and Lactobacilli. The properties that these organisms seem to share in common are the ability to produce acid from dietary carbohydrates. Most importantly, the ability to tolerate the acid they make, because many organisms in fact can make acid, but few of them actually like it when they've done it. I was going to say like me a drink, but perhaps that's not a good metaphor. And they produce extracellular, intracellular polysaccharides from sucrose. In periodontal diseases, we get a different flora. The flora seems to shift in a totally different direction. And we get increased numbers of gram-negative anaerobes, nasty names, Porphyromonas gingivalis, Actinobacillus, Actinomycetum contaminated Agriacibacteria, Actinomycetum. We get spirochetes, we get Anarella, and the unculturables. And they seem to be associated with the production of proteases that can degrade the host compounds in the crevice, cytotoxins that damage the host cells there. They induce an inflammatory response, But the trick is the inflammatory response is supposed to be helpful to us to control the organism. What some of these organisms do, their proteases, is actually break down the immune modulators. The backup defense that the host has to dampen the inflammatory response gets broken down. So you get an uncontrolled, inappropriate inflammatory response. And that in itself causes bystander damage to the tissues. So the thing is, if you're a microbiologist and you haven't got a nice yacht and you're not going on a golfing holiday, you lie awake at night and you wonder, why? Well, not why I've got a yacht, but why does it happen? How did it happen? And what can you do to stop it and reverse it? Now, one of the key questions is, where does pathogens come from? Because when you're old, like I am, you've seen various things come and probably go. And one thing was whether dental diseases, and it's a reasonable question, could be regarded as exogenous diseases. You acquired the organism from someone, and then you established an infection, just like many other diseases. Certainly in the early days, if you tried to look for some of these periodontal organisms with the limits of culture that we had, it was very difficult to detect them. But now we have molecular techniques like PCR, where you amplify small bits of DNA, and the key word is amplify. Even if they're at very low levels, you can detect them. And this is checkerboard, another molecular system for detecting organisms without growing them. People found that actually many of these organisms that have been implicated in disease are there in health. Now, it might mean that these people would subsequently go down with disease at some time in the future. These are kind of cross-sectional studies. Or you could interpret the fact that these organisms are present naturally most of the time, but at levels that are so low that they're not clinically relevant. You can get transmission, certainly from mother to child with mutant streptococci, and between spouses for some of the periodontal organisms, which I think is a business opportunity for them, because when someone lines up a partner, perhaps I could check their periodontal pathogens from before they engage in anything where they might be transferring organisms between one another. If anyone wants to talk to me about that, I can be found later. So, to my mind, this is where we simplify things down. Very complex. We've got loads of organisms there. But if we think back to health, we know these organisms are present. And if we look at health here, the green ones are what we have naturally and giving us benefit. One situation is that we have very low levels, shown in red there, of the organisms that we might implicate in disease. So they're there already, the levels are so low that they're not clinically relevant. It's like carriage of an organism. It could be they're not there at all, that's one theory, and we acquire them, so they're transmitted to us. But in either case, for disease to occur, where we know when we take samples of back from disease sites, you get elevated levels of these nasty red organisms. For that to happen, they've got to outcompete the green organisms. And that shouldn't happen from ecological principles. The only way that can happen is for some change, some major ecological pressure that suddenly alters competitiveness of the organisms. And most likely changes are a change in the diet, increase in inflammation at the site, a reduction in the host defenses, and a change in local pH. So what therefore we need to do is to interfere with those changes and push the system back in the opposite direction. So I'm now going to just go through very quickly dynamic relationship between the environment and bacteria. So we know from looking at microbial consortia that are in ponds, in the gut, in the mouth, that the sort of factors that determine whether you change the flora or not are like how much you get different food there, because there are organisms that require certain different food. If pH changes, or if there's a change in the status of the host response, or whether it becomes more aerobic. So the key one in the mouth is obviously, do we get more fermentable sugars in the diet more regularly? Because that will also change pH. And in caries, we get an acid stress, we get low pH in plaque. Other organisms are not going to like that. Subgingially, we get a small rise in pH, so it can become alkaline there. And again, that will favor the growth of some organisms, particularly when they're metabolizing gingival crevicular fluid. It's obviously a problem if your host defenses are compromised, whether it's through neutrophil defects or a reduction in saliva flow, and that can occur through all sorts of factors like taking medication for an unrelated problem. Likewise, when plaque builds up, it can get very anaerobic, and that can encourage the growth of some of the anaerobic pathogens. Bacteria are what's known as plastic. They will respond. They're not made of plastic, but they are plastic in terms of their response. They are not constant in what genes they express or what properties they exhibit. So when environment changes, they shift the gene expression and their properties accordingly. So if we grow an organism in pure culture and look at the effect of pH or sugar, we can measure properties like, you get more acid production, less acid production, more proteases, less proteases. And we initially spent a lot of time trying to explore this. And we used a system called a chemostat. This is again used in general microbiology. You grow your bacteria in a vessel, but you can control the conditions, whether it's temperature, pH, oxygen, and composition of the media. And it's very precise. You can grow the organisms indefinitely under a standard set of conditions and say, for this set of condition, the organism does this. If change the amount of sugar there, they do that. And that's where we start from. So we know the environment changes during disease. We know oral bacteria are capable of responding to these environmental changes. And we look to see at the sort of environmental changes we see in disease, how that affects the so-called pathogenic organisms. And summarizing a lot of information, we found that organisms that we associate with caries, like mutans, streptococci, and lactobacilli, grow better at low pH and on high sugar. They make more acid, particularly more lactic acid, and the growth is actually optimum below 7. In contrast, if you take a periodontal organism like Biofilm gingivalis, it regulates its proteases at alkaline conditions. And when there's lots of hemin, it needs hemin for growth. It binds hemin in the mouth and the breakdown of gingival crevicular fluid that has hemoglobin, haptoglobin, and other heme-containing molecules. Again, its growth optimum is more in the alkaline side of things. And under these conditions, under these disease-related conditions, we see upregulation in genes that are associated with virulence. So we studied, for example, organisms implicated in caries, strep mutans, and compared it to organisms that are not, that are implicated in health. And we show that there are differences. Under some conditions like lots of sugar or at low pH, strep mutans did more. Grew faster, made more acid. And likewise, when we took a periodontal organism and grew it when there's lots of hemin, which would predict to be the case in inflammation in some gingival crevice, it upregulated some of its nasty proteases, can be very destructive to tissues and break down the host control proteins. Well, that was okay for a while, kept me in business for a while. Then the big jump was to say, but we're studying organisms by definition. Pasteur himself had done. I said I was old. I'm not that old. It was only by reading his work. You study organisms in isolation. So that's the only way you understand what that organism is capable of doing. But as I stressed, plaque is a community of organisms. And now we're wiser. We realize the properties of a mixture of organisms can be different to just adding up properties of individuals. It's more than a sum. So we began to realize that something that happens to this organism may affect its relationship with that organism. So although it was contrary to dogma, we started to grow organisms together. That's just not the thing you should do. Because we could see the effect of a change in a key environmental parameter on the competitiveness of the organism and how that might affect the structure of the microbial community. We initially started with dental plaque, seemed obvious, but when we started to grow dental plaque as mixtures, just like studying patients where they have all the organisms, so we suddenly found organisms we didn't know were there in the first place. We didn't know where they'd come from. We couldn't replicate the study because we couldn't manipulate the composition. So we decided to go to an in-between stage where we had 9 or 10 organisms that were either found in health or caries or perio and grew them together. So we knew they were there at the beginning. We chose them because we were lazy. We could easily identify them. They had some factor that allowed us to identify them. We could store these mixtures in a freezer, and then we could bring them out in record experiments. So we had good reproducibility, and we could take one out or put an extra one in if we had experimental question. The big trick was to use a medium to grow them in that reflected habitat. So we used something that had mucin, glycoproteins in. And we used the hemostat device because we could grow the organisms indefinitely for weeks at a time under standard conditions, and then we could change one parameter at a time. We could change pH without changing growth rate, without changing the concentration of sugars. So we could get true cause and effect relationships. We could introduce surfaces and develop slightly more complex systems, which I'll show you very quickly in some slides. It's not really important for you to understand. So this is the sort of system we have. This is our hemostat, a vessel grow bacteria in, and we can control most of the parameters and vary them independently. For some experiments, so we feed it that medium, we could put this stuff into a second stage hemostat, another one, and mess that one up without affecting this, because it takes a long time to do. And we put surfaces in there so we could grow biofilms. And then for another application, we'd feed that into a different biofilm device where we could grow biofilms at different depths, thin ones, thick ones, and so on. This is this biofilm depth device. It looks like it's got a rotating turntable. Those that have long playing records would know what the significance of a turntable is. The biofilms grow in there, and you can recess little plugs in there to a particular depth. So you can have them to any defined depth. These are the organisms. We've got some that are associated with health, caries, and periodontal disease. As I said, one of the first key steps was to grow it on mucin. And this is what a mucin molecule would look like, a protein backbone and side chains with sugars. For the organism to use these sugars, and this is just a tip, if you get reincarnated as an oral bacterium, it may be the only sugar you can use is a lactose, but you can't get to it because there's a sialic acid on the end. You need to find someone who takes off that sialic acid. Okay, just free tip. So the organisms that we had, we selected, some had very little ability to break down these mucins. Others had very specific major enzyme activities. So we put 5 of the ones that could do hardly anything together in this immunity, and they grew to these sort of numbers of organisms. 5 of them together, that's their initials, doesn't really matter. We then add in the 2 bacteria that had silidase. They will take off that sialic acid, and they grew up. And in fact, what yellow means is very complicated to explain, is that the numbers of these organisms weren't very big, and they're hidden in between there as blue. But what the effect of adding in sialidase, it had a boost to those that were already there, because some of those wanted galactose. So you get an increase in the ones that were 5 that were already there, went up, shown by this yellow bar. We added in someone who could take off fucose, lactobacillus, and again, you can't see its numbers because it's hidden as a blue bar there. But it also had bonus on those that were already there, another boost with the yellow bar. And perhaps you'll see here clearly, when we add in a big protease juicer, you can see the numbers of the protease juicer itself, but again, because that suddenly releases peptide and amino acids for the others, you get an increase in those that are already there in the yellow. When we added another organism, 10, it had nothing new to offer, so nothing really happened. So these organisms are acting as a true community. They're interacting, they're dependent on one another, and that was important. So what they're doing, we had one organism that takes this, takes that, that means organism 3 can have a go, that means the protein is now available for organism 4, and so on. The other thing is, how else are they functioning together as a community? And these organisms, these are what are called facultatively anaerobic, the green ones. They can go in the presence or absence of oxygen. The orange one is aerobic, likes oxygen, and the red are anaerobic. So we were growing this system where we were making anaerobic. So in the inoculum, in the first stage of a 2-stage vessel, the anaerobes dominate. The anaerobes are in charge of anaerobic conditions. The aerobe is very detectable. We fed this into a second vessel where we put in as much air as possible, which is my mouth at the moment. There's a lot of air going through it. And to our surprise, the anaerobes are still there. The aerobe became dominant because it's suddenly all that oxygen that it needs for its metabolism. But somehow those anaerobes that shouldn't be going in this situation are there. And even if we look in the liquid environment, because we thought there might be a biofilm effect, same situation. When we measured the amount of oxygen there, couldn't detect it, less than 1%, even though we're pumping in air as fast as a pump will pump. And in fact, this measure of anaerobiosis means it's very anaerobic. And we concluded that these function as a true community. It's the aerobe, the Neisseria, that no one has hardly looked at all, but found in all plaque sample, is consuming all the oxygen in that system, which means anaerobes flourish. So we tricked the culture, we took out Neisseria, so now we knew that the anaerobes wouldn't grow in an aerobic situation. But below us down, the microbiologists, the anaerobes were still there, and this time it was the streptococci that were using the oxygen not as effectively. We did get oxygen there. We proved how smart you think you are. Anaerobes don't grow in the presence of oxygen. We all know that. They do, and they grow as a community. And it just shows how these organisms interact. We even tested that they were true anaerobes. We measured how long they could survive on their own in air, and it's in minutes. We thought perhaps it's because they aggregate with one another. We put the anaerobe with aerobe. No one ever looked for coagulation with an aerobic organism like Neisseria. And the only evidence, this is a numerical score where 4 is a lot, and 0 to 1 hardly anything, was with Fusebacterium. And Fusebacterium will coagulate with everything. So when we add the Fusebacteria in, we did see improved coagulation. So we thought that perhaps in surviving the oxygen, the organisms forming functional units. They're forming a consortium that can cope with this. So this is Neisseria and an anaerobe, and there's very small little aggregates there. When we add the Fusebacterium in, we get these big consortia, big units, and our inference is in here. There's very little oxygen. This just shows that when we look at the numbers of these 2 anaerobic organisms, when the Fusebacterium is there, we get substantial portions of them. When it's not, we find hardly any when we aerate, pumping oxygen into the community. So we were quite confident that our consortium could form biofilm and it functioned as a true community. So now go back to this. Why do we get these shifts? We could set up a culture where we have health and try and shift, find out factors that would drive it towards caries or drive it towards periodontal disease. We know caries is associated with lots of sugar and low pH, and periodontal diseases with increases gingival clavicular fluid and raises pH. So we explored those. So we took our experimental system, 10 species, and the design initially, this is a long time ago, young man, dark hair, we pulsed in a fermentable sugar, which shows glucose for simplicity, each day, once a day, because the kinetics of the system would be that glucose would be lost, ready for next day when we had a fresh pulse. But we could exploit the unique capability of this system that we can keep the pH constant at neutral pH, and we just see the effect of sugar per se. There is no other model system you can do this. If you add it to a human or an animal, as soon as you add in sugar, the pH changes. You can never work out whether it's pH or sugar that does this. As the control, we add in the glucose and we let pH find its own level through bacterial metabolism. So before we did pulsing, what we found with high levels of the healthy bacteria, low levels of mutans streptococci, and low levels of lactobacilli. They're less than 1% combined of the total flora. When we did the study with the glucose pulse at constant pH 7, the flora was relatively unchanged. Even though there was fermentable sugar there, the organisms did not benefit from it. They stayed at less than 1% of the flora. However, when we changed the system and let the pH change following the glucose pulse, gradually over the 10 successive days, we got dominance of the mutans streptococci and lactobacilli, and the health species were reduced. This is a moderately good slide. I scanned this at home just before I came away, and at home I've got a newer version of PowerPoint and I've got work. I just could not get this to go. Of course, what I did, I got my son to do it for me. That's when you know you're getting old. So this is from the original paper, and what we have here, it's not as clear as I'd like it, but the squares, if we look at this sanguineous, which is associated with health, an organism associated with health, the squares are we pulse with glucose on 10 occasions. It shows 10 days, and it shows the vial counts, but the pH is kept constant at pH 7. And this lower line, the cross, is where there's no pH control. The culture of the 10 organisms, every time it's the glucose could make however much acid wanted 6 hours, then we bring the pH back, leave it overnight, give it a pulse the next day. And you can see it tolerates the first few pulses, but then we get this oscillation. Each time it pulsed, the pH went further, went lower, and had more and more of a negative effect. These are numbers, log numbers, bacteria. So at the end of 10 pulse, it was almost non-detectable. And this can be contrasted to strep mutans, which is implicated in care, obviously. And again, the square is the case where it stops off, and we've got no pulsing. And as we pulse, the pH is kept constant, it pretty much stays at the same number of cells. But when we do with crosses, we have no pH control. It again gradually dominates within the culture. And if the bottom graph shows the amount of acid produced after each pulse, and that's the square, you can see this is a log hydrogen ion concentration. After each pulse, we get more and more acid. We select a little bit of extra strep mutans, a little bit of lactobacilli, next pulse comes in, they make even more acid. They grow up because they love low pH conditions, so the next pulse comes in, they make even more acid. And so this is the amount of acid they made, and this triangle is the time to reach an arbitrary pH 5. Again, the time reduces. The amount of acid made increases, the time they take to do it gets shorter. So you're getting a more and more catastrophic effect on the flora. And if we then went on to study this by stopping the experiment at a particular pH, so the bacteria could make acid, but we would only let them make so much. So we set up a whole independent experiment where we let the pH go to 7, no change at all, or to 5.5, set a totally new experiment and it could go to 5, set up a totally new experiment, a pH go to 4.5, this is what we had for the new pH control. And this is for two organisms that like acidic conditions, and you can see there is almost a dose-response curve. This is the terminal, the results after the final pulse, and you can see that the lower the pH, the greater the proportion of the strep mutans or lactobacilli in the culture. And if we look at two organisms that are very pH sensitive, we see it goes the opposite direction. And this was supported, or you could find evidence in literature. This is known as Svanberg, Sweden, something done in 1980, where she got patient volunteers to rinse with phosphate buffer of different pHs, and just look for the percentage mutans streptococci in black, and you can see that when you had 2 lots of rinsing with low pH buffer, you again select out mutans streptococci in the black. So the conclusion of this was it's the low pH rather than just sugar availability per se that selects carcinogenic bacteria. And there's a relationship. The lower the pH, the higher levels of carcinogenic organism. And again, it posed the question, could inhibitors of acid production prevent this selection? So it shows fluoride. At the time, fluoride was obviously recognized as a very important molecule, but people didn't really felt it could have an effect on the all organisms, because MIC, sensitivity for the growth of an organism at pH 7, fluoride means you'd have to have massive concentrations, which would never achieved in plaque in humans. Sub-lethal levels of fluoride interfere with enzymes within the bacteria, and certainly it was known in mutans streptococci, fluoride penetrated and affected these enzymes, preferentially at low pH. It was perfect. As pH starts to fall, the fluoride goes in and inhibits the bacteria. So we did this in a number of different systems where we had moderately low levels of fluoride. People can argue whether these are realistic or not. So again, it's the same setup. We saw effects of glucose pulsing, let pH change, effects of glucose pulsing, let the pH change, but we added in fluoride. And what we saw, as before, if you don't let pH change, mutans, streptococci, and lactobacilli don't respond. If you let the pH change, they get dominated by those organisms. If you add fluoride, the flora is again relatively unchanged. The mutans, streptococci, and the lactobacilli are unable to exploit a set of conditions that you would think they would be able to. And if you look at the levels of S mutans, in this particular study, before we started the pulsing, there were 4% of the community of organisms. If glucose pulse without, with fluoride, we got up to 23%. If we had the fluoride there, they stayed around 3% of the culture. And again, there was a difference in the pH. And again, you can see this hydrogen ion concentration. With just glucose alone, you get a lot of protons produced, and you have the glucose plus fluoride. That ability to produce acid is retarded. And this is the time to reach pH 5. And again, when you've got fluoride there, you slow the rate of acid production. We did this more recently in biofilms, because that was done in planktonic cultures, and biofilm people obviously say this because you've done it like that. And this is where we've taken a section in a biofilm developed in that constant depth thermometer. And this time we've moved on from with that pH system we had before. When having a grayscale, we have a color scale now, where pH is around neutrality are yellow and orange. pHs that are acidic are the hotter colors, red and through to blue. And this is time sequence in minutes, looking at the same section of the black biofilm at a particular depth within that. We know what the organisms are. It's a mixed culture of organisms. And you can see as time goes by, with glucose alone, it gets hotter. You get more and more acid produced, and that can be converted into a normal pH 4 curve. When we do it with low levels of fluoride, colors stay much more in the yellow and light orange, and pH 4 isn't as far. And in fact, we got quite a substantial reduction in pH change in this artificial biofilm. So fluoride can have an antimicrobial effect because it can directly inhibit bacterial enzymes associated with sugar metabolism and pH tolerance. It interferes at a number of levels. But it also has an indirect effect, because if it's not making much acid, if it's not changing the pH, it's taking away the most major competitive environmental factor that mutant streptococci exploit. They grow up because the conditions get acidic. That's their optimum. That's what they like best. In the Olympics, they do best at acidic conditions. It's not getting to those acidic conditions because of the direct inhibition effects. They're also unable to exploit the situation even with sugar present. So it works at two levels.
What do you think the mechanism of fluoride is going in terms of?
Well, it can inhibit enolase, which is in the glycolytic pathway where glucose goes to be made to lactic acid. It can also interfere. It goes in as HF. At low pH, the fluoride becomes HF, goes into the cell. The fluoride goes off and inhibits certain enzymes. But then there's protons inside, which the organism doesn't like. It acidifies the inside, and the organ has to spend energy pumping out those protons. So it's sort of weakening the metabolism of the cell. So it can work at quite a number of subtle levels that cumulatively make strep mutans not like fluoride. You could never design a molecule like fluoride to do all these things. And we also looked at some sugar substitutes, because at the time they were being proposed, and found that xylitol prevented the selection of mutans streptococci, and glucose was present. It reached around 20% of the flora. Curiously, when we had glucose sorbitol, it seemed to like that. But when we had glucose plus xylitol, it really didn't like the combination. At the time, the theory was xylitol goes to the cell. Again, the organism has to spend energy pumping that xylitol out. And the xylitol goes into what's called a futile pathway that blocks the metabolism of the organism. Some people dispute this, but we just did the experiment and those are the results. So I said the low pH rather than carbohydrate availability sex for carcinogenic bacteria. There's a relationship between how low the pH goes and how high we get carcinogenic organisms, and the inhibitive acid production can prevent selection of carcinogenic bacteria. We've done a little bit of work on periodontal disease, interested again in gingival crevicular fluid as not only a source of the host defenses, but also as providing novel nutrients. And these experiments, serum has been used as a surrogate for gingival crevicular fluid because we need quite a lot of it for these experimental systems. What had given us encouragement, well, again, a study, before some of you were born, I expect, done in the Netherlands, where a Dutch group had taken samples of plaque from patients, 3 samples of plaque, in which they looked for organisms at that time was considered relatively important in periodontal disease, Prevotella intermedia. And in the sample of black, 2 of them they couldn't detect it, ND is not detected, and in the third one it was less than 1% of the flora, so not really important. They then did what's called enrichment cultures, where they would put the plaque in some serum for a few days, let it grow up, and then look for the target organism. And again, after 1 enrichment, they couldn't detect it. At low levels, couldn't detect it. By enrichment step 2, they could detect quite a lot of it in the first sample, but not in the other 2. They then put it into fresh serum, and in enrichment step 3, it was detected in all 3 of them. You can see as they go on through the enrichments, you could suddenly detect the organism, and it reached quite high levels. This means that that organism is present, but at levels that were below the detection limit. You change the nutrient source, and suddenly it's a competitive organism. It's there in large numbers. It shows you can push the composition of the black flora just by changing key parameters. So we've done a study with our same community, our same very defined system, and we grew it in medium. 10 organisms there. It was highly anaerobic. That's a very, very low level of redox potential. And the pH was around pH 7, which is what you find in a healthy ginger or crevice. When we measured protease activity, it was low. When we measured numbers of Porphyromonas gingivalis, it was low. At this point, we added in serum as a surrogate for gingival orbicular fluid and let culture do what it wanted to. What we found was the pH rose. It went up to pH 7.5, which is the sort of pH used to find in inflamed periodontal pockets. It became even more anaerobic. The redox potential went lower. And the protease activity went really high. And Porphyromonas gingivalis became almost 80% of the composition of that community. So again, simply by changing the nutrient source, we distorted the composition of that community. We did another study where we grew just 3 organisms that are all called black pigmented anaerobes. They make a black pigment, very easy to see. Porphyromonas gingivalis found in advanced periodontal disease. This may have a role. It's in the orange complex. This is found more in healthy plaque. These 2 pHs are what people might find in a healthy gingival crevice. What we found when we grew these 3 organisms together, those 2 organisms, that organism was the dominant organism out of the 3. We just shift the pH of that culture by a quarter of a unit, and suddenly Prevotella intermedia is more competitive, and we get significant levels of Porphyromonas gingivalis. Shift another quarter of a unit, which is what can be found in inflamed pockets, and this now is dominating. It's far more competitive than the organism we found with health. So again, we see just changing nutrient status, the pH, you can shift. These organisms are very responsive, shift the composition of the flora. So we had these 3 principles earlier. You can see that the relationship between the organisms and the environment is active and dynamic, and it probably can be manipulated. So just finishing off, I don't know about you, but I'm just about parched. The theories that have been put forward to explain the relationship of plaque to disease for a long time were the specific plaque hypothesis put forward by one of the most distinguished oral microbiologists, Walter Lowy. And this is really very helpful. Thank you. Thank you. Because instead of looking at all the organisms that we know are in plaque and see and wondering, well, which one of them is it, from a series of studies, he said, well, Strep mutans, Strep sabinus are very clearly associated with caries. We must focus on those. So that helped us to suddenly start to understand these organisms. There was a different view, perhaps come more from Europe, which was the non-specific plaque hypothesis, and it was more applied to periodontal disease, which said, but all the organisms have some role. Some are bad for us, some are actually good for us, and it's the net outcome of all those interactions that's important. So we should concentrate and know all of the organisms. Sometimes we'd hold conferences on this. People would almost be fighting as which camp you fell in. If the diseases weren't specific, they certainly showed evidence of specificity. When you look at disease sites, you certainly see certain groups of organisms consistently there. And I felt what was important factor missing was what selected for these organisms. Because I was coming to the conclusion at that time that they were there. It was more imbalance in what we had that led to disease. So to get that environmental factor in, to reconcile the sort of fairly subtle differences between both of those hypotheses, I put forward the ecological plaque hypothesis. And the benefit of it, I think, was that etiology need not be monospecific. So many organisms could contribute to it. It didn't worry me that it wasn't just a single organism. You could carry so-called pathogens. You know, it wasn't a problem that some people found when you started finding disease sites without pathogen or healthy sites that had the pathogen. It's well known you can carry these organisms. And none of the factors that we're talking about in disease are so specific. It's not like a specific toxin. Clostridium tetanus that causes tetanus produces a specific toxin. If you have that toxin, give it to a person, they'll suffer from tetanus. We're talking acid production that other bacteria make. The key thing for me was that disease could be prevented or controlled not only by directly inhibiting the cause of organisms with all the approaches that were being put forward by the specific hypothesis, but the key thing was you need to interfere with the factors that were driving these deleterious shifts. Because if you didn't interfere with those driving factors, the patient would only come back in a few months' time with the disease again. You hadn't eliminated the problem. All you'd done was dealt with it that time. So in carriers, what we got was if you took in sugar, acid was made. This causes a stress. pH shifts from neutral to low pH. We get an environmental shift. And we get a shift from organisms like sanguinis and gordonii. We find health in mutant streptococci and lactobacilli. So we get this ecological shift. And we tend to push the system more towards health and carriers. My argument is, yes, you can deal with these organisms directly, but unless you interfere with the factors that are driving the emergence of these organisms, you won't be totally dealing with the problem. So what we have is that the environment is selecting factors with properties that are both acid-producing and acid-tolerating. And while these traits are associated with mutant streptococci, they're not uniquely so. So people have found other acid-tolerating streps that are not in the mutant streptococci group. That's fine. It's no problem to me at all. Likewise, there are other streps that on their own can produce lots of acid, as much as mutant streptococci. Well, of course they'll be important. You know, we're not looking for a specific organism. And this is shown here because it's now recognized that not all mutant streptococci are equally acid tolerant, and that several streptococcal species can behave similarly. It's just those traits are most constantly focused with mutant streptococci, but they're not exclusively so. Likewise, if you look at bacteria that can grow at low pH, quite a few organisms can. It's not exquisitely associated with mutant streptococci. Again, the velocity of acid production by some strains at particular pHs are equivalent or better than mutant streptococci. So a lot of organisms will play a role. So the way you could try and reduce the sugar flow or low pH challenge is obviously through fluoride, clearly through dietary control, reducing the frequency of providing fermentable sugars, encouraging the use of products that have non-fermentable substrates in sugar substitutes, stimulating where possible saliva flow, and antimicrobial agents in dental products. Perhaps in Europe we have far more of these with metal salts and triglycerides. Even at sub-MSC levels, these will interfere with sugar transport and acid production, even if they don't actually kill the organisms. And as I have already alluded to, the use of products that contain sweeteners can't be metabolized by the oral flora will have benefit because they'll stimulate saliva flow and the benefits of saliva flow without leading to damaging amount of acid. If we look at gingivitis, it's a bit more complicated, but when plaque accumulates, we get a stress, we get increased inflammation. That's going to cause an environmental change. We're going to get increases in gingival curricular fluid flow. We're going to get bleeding, a raised pH and temperature, and this tends to drive a shift in the balance of the organisms. We'll shift from a gram-positive flora to great numbers of gram-negative organisms, obligate anaerobes. This will tend to push the system more towards gingivitis or periodontal disease and away from health. This system is selecting for those bacteria with properties that are proteolytic and like to grow under slightly alkaline conditions. And these traits, again, are associated with organisms that we commonly find there, like Porphyromonas gingivalis, T4 Cyphia and spirochetes, but not uniquely so. And again, the way one can try and alter the gingival environment is with oxygenating or redox agents. People are looking at molecules that can shift redox potential to make it less anaerobic, to make it less favorable to the growth of anaerobic organisms, and to reduce inflammation and gingival curricular fluid flow. So people are beginning to develop different classes of anti-inflammatory agents. And again, I should say that some antimicrobial agents like chlorhexidine, triclosan, metal salt, even though not killing the organism, can often inhibit the proteases these organisms use to cause their damage. So much favoured in dental communities is the seesaw or the balance. So just to remind us that this is a dynamic and active relationship. That the microbe interactions, host microbe interactions are delicately poised. And when they're poised appropriately, we get benefit and harmony from this relationship. At times, if we have an inappropriate diet, all hygiene is rubbish, host defense is suppressed, that just shifts the balance and we get a problem with this host microbe interaction and disease occurs. I think, therefore, which I'm so impressed with this group, and it's been so uplifting for me to hear people talk, is this holistic approach to dental disease, because it's absolutely true. I spent life looking at plaque and so on. It's a little translation of some of these ideas to dental disease. And if you go to conferences, people talking about saliva, the answer is all with saliva, or it's diet and lifestyle, or host defense people. Some microbiologists look to think they got the answer. And really, all these are interconnected. Your diet affects the microflora. The lifestyle can affect saliva flow, which again will affect the microflora, and so on and so forth. So you need to take a far more holistic approach to understanding what may be going wrong. Is that the question?
Have you ever had where
where we bring things back from, well, we haven't done that particularly, but I think that's what people are looking at, and that's what they're saying they can do. So you could take someone where the conditions are inappropriate, perhaps they already have disease or they're susceptible to disease, and the feeling is you could apply specific approaches when you identify what it is their particular risk factor and try and bring them back within the boundaries of what may be called normal. So just to finish off, I've been very patient. To emphasize that dental plaque is both biofilm and microbial community, that it's natural and beneficial to health, it does good things for us, but the plaque bacteria respond to environmental changes. That the plaque-mediated diseases seem to be due to changes in the local environment, which can then enrich organisms that were only a minor component of the plaque consortium. So if dental diseases aren't exactly ecological catastrophes, perhaps they're micro-ecological catastrophes. And I think the ecological concept does offer novel therapeutic possibilities, and particularly also educational and communication opportunities. People are very aware of the impact of the environment on their lifestyle, and you can translate that down to events that are occurring in their own mouth. So I'd like to thank you for your attention. I haven't gone too long, and I'm here the whole conference, so if people want to talk to me about things, I'm more than happy to do so. Thank you very much.
Professor Marsh, on behalf of Congress, I'd like to thank you very much for an excellent presentation. We've been looking forward to it. We're not turning that down. Done now.
Okay. Thank you.
Professor Marsh,
Thank
on
you.
behalf of the Awards Committee, we have one more accolade to add to your long list of accolades. You've been presented with a Lifetime Achievement Award from our organization.
Thank you very much. I'm very grateful for this. Thank you very much.